Hello students, in this part we will discuss the Mendel's monohybrid, dihybrid and trihybrid phenotypically and genotypically ratio. How we can calculate the phenotypic and genotypic ratio in monohybrid, dihybrid and trihybrid. Mendel's crossing process. Mendel crossed one pair of traits with each other, two pairs of traits with each other, and three pairs of traits with each other. One pair of traits is known as monohybrid, two pairs of traits dihybrid, three pairs of traits trihybrid. We have discussed monohybrid cross. Monohybrid cross. Mendel crossed one pair of traits with each other, such like a tall plant with a dwarf plant. Tall plants are represented by one pair of genes, capital T, and dwarf, small t. When he crossed in F1 generation, he saw that all plants are tall. In F1 generation, he saw that all plants are tall. He again crossed F1 generation, means interspecific cross. कैपिटल टी एंड स्मॉल टी विद कैपिटल टी एंड स्मॉल टी F1 जनरेशन का दोबारा क्रॉसिंग रही. इन F2 जनरेशन ही saw that F2 generation or F2 crossing he saw that such like a cross are obtained, means three plants are tall and one plants remain small. So phenotype ratio of monohybrid cross. Phenotype ratio means there are three plants are tall 3, and one plant is dwarf. Means there is 3:1, that is the phenotype ratio of monohybrid cross. And what is the genotype ratio? Genotype ratio of monohybrid cross, that is this one pair is homo and this one pair is homo and these two pairs are hetero. So the genotype ratio is 1:2:1. Again, this is the monohybrid phenotype and genotype ratio. Then he crossed two pairs of traits with each other. Two pairs of traits, that is called dihybrid cross. टू पेयर्स ऑफ ट्रेट्स आपस में क्रॉस रहे हैं, तो उसका फिनोटाइप और टू डाइहाइब्रिड रेशियो क्या होगा? फिनोटाइप रेशियो ऑफ मोनोहाइब्रिड क्या था? 3:1, that is number of 1, means one pair of traits. Then he crossed two pairs of traits. So how we can calculate the phenotype and genotype ratio? We can say that monohybrid cross ratio is 3:1, that is the one pair of traits. Now in this he crossed two pairs of traits, means 3:1 the whole square. How we can calculate? How we can represent 3:1 into 3:1? Clear? By representation the phenotype ratio of dihybrid cross. Crossing process we can make 3 multiply with 3, 9, 3 1 jar 3, then 1 3, 3 1 jar 3, and then 1 1 jar. This is the dihybrid cross phenotype ratio. How we calculate? We can calculate a simpler method. 3:1 in one pair of traits, then we can power the 1. Dihybrid cross, we can square it, and just like as we can calculate the genotype ratio. Genotype ratio of monohybrid is 1:2:1. 1:2:1. While he crossed one pair of traits, so it means power 1. But in this case is two pairs of traits, we can calculate it double time. So how we calculate? 1:2:1 into 1:2:1. We can multiply it. 1, then 2, then 1. We can multiply with 1. Then now the number is 2. 2 1s are 2, 2 2s are 4, and 2 1s are 2. Clear? Now it is the ratio of it is multiplied with 1. 1:1, 2, and 1. So while crossing Xenotype ratio in dihybrid we can calculate easily. We have obtained in monohybrid xenotype ratio is 1:2:1, while he grows one pair of traits. And while he grows dihybrid means two pairs of traits. We can multiply it by the double times. So in genotype ratio dihybrid growth is 1:2:1:2:4, and then 2:1 and 2:1. This is the dihybrid growth genotype ratio, and this one is the phenotype ratio. Then Mendel grows three pairs of traits, and that is called trihybrid growth. Trihybrid means three pairs of traits. What is the ratio of phenotype and genotype of monohybrid? Monohybrid phenotype is 3:1 and genotype is 1:2:1. तो मोनोहाइब्रिड का चेक कीजिए पहले. मोनोहाइब्रिड phenotype that is 3:1. It means while he grows one pair of traits, and genotype while he grows one pair of traits, the genotype ratio is 1:2:1. One pair of traits. Now he grows three pairs of traits, so we can calculate by the 3 times phenotype ratio of trihybrid growth. 3:1. This is the monohybrid growth ratio, phenotype ratio. Now it is trihybrid, so we can calculate 3 times. We have obtained 2 times ratio. Phenotype कितना था? 9:3:1, and we can also write it 3:1 3 times. 3:1 3:1. Clear? The multiplication of the 3:1 into 3:1 is this one, like 9:3:1. This is the multiplication of these two, and again 3:1 third time. We can obtain 3 9s are 27, 3 3s are 9, 3 3s are 9, 3. Now we can multiply with 1 with all these. We can obtain 9, 3:3, 3 and 1. We can arrange it 27:9:9:9:3:3:1. These alls are in trihybrid growth, that is the polygenic. We can obtain nearly 64, and this is the phenotype ratio of trihybrid growth. We can multiply it 3 times of the monohybrid growth ratio. And trihybrid growth genotype ratio of trihybrid. What is the monohybrid genotype ratio? 1:2:1. Clear? And now it is trihybrid. We can multiply with 3 times. We can write it 1:2:1 into 1:2:1, and again 1:2:1. Clear? The multiplication of these two as on this one. This क्या? 1:2:4:2:1:2:1. This is the multiplication of both twos. And again we can multiply it 1:2:1. Multiply 1 with all these. 1:2:1, 4, 2:1, 2:1. This is the multiplication of 1 with all these. Now multiply 2 with all these. 2:4, 2, 4, 8, 4, 2, 4, 2, and 4 and 2. This is the multiplication of 2 by all these. And now again multiply 1 with all these. Then we can obtain 1:2:1:4:2:1:2:1. We can calculate these are the 64 progeny. 1, 3, 4, 6, 10, 12, 13, 15, 16. 16 into 8, 18, 22, 24, 28, 36. 40, 42, 42 and 46, 48, 49, 51, 52, 54, 58, 60, 61, 63 and 64. These are 64 progeny in trihybrid growth. So we can calculate the phenotype and genotype ratio of monohybrid growth, dihybrid growth and trihybrid growth, such like this formula. So Mendel used mathematical and statistical formula. This was the disadvantage at that time because this was a new concept for the scientists at that time. So the work of Mendel's take a longer time to limelight in the public because at the contemporary of Mendel's, Robert Darwin and Wallace didn't accept these mathematically formula, but now it is very easy to express these expressions by the monohybrid growth, dihybrid growth and trihybrid growth, genotype and phenotype ratio.